it all builds on what we started with. So like I'm going to trust you. I won't go back and review A pairs with T, G pairs, like that basic stuff. But let's go back at least to the beginning of gene expression. I know, but I can't because it's a lot. And you already know A and T genes. Curvy things go with straight things, right? Let's start with transcription. Yeah. Whoa. Okay. Well, do you want to go back to replication? We can do that. So replication was kind of training wheels for transcription, right? Replication was, okay, we've got DNA that gets split with an enzyme called helicase, like so. And then 2 copies of the DNA polymerase enzyme run along each split strand and match up, because the shapes work, they match up the right bases to the right strands. So we get these 2 identical, but only really half new, half old strands of DNA. Good? Questions? Also, it happens a bazillion places at once in all that 6 feet of DNA, so you get it done in a reasonable amount of time. But so to bring in RNA in gene expression, we've got these 2 phases: transcription, which is kind of like replication, but only on 1 strand, and translation, which is where all the moving parts come. This is all to make proteins, which are important because they do almost everything in your body, like hold you together, like make chemical reactions happen and all that. So transcription, you split the DNA this time and add the complementary RNA this time with the same enzyme, which is called RNA polymerase, making a single strand of messenger RNA, which then peels off and hangs out by itself and leaves the nucleus, but not before it gets edited, taking out introns, splicing back together exons, for example, that. And some of you are going to ask, I know, well, how do you tell which things are going to come out and which things are the introns and which things are the exons? You can't. But your cell knows, right? So, we'll always tell you that whether this is the finished RNA or whether it's unedited. But once it's edited, it then gets read in 3-letter segments called codons. And this is the point where we can stop and cheat, right? This is the point where we can use the decoder wheel. And we can go straight from mRNA to amino acids. Again, the classic mistake is to accidentally use tRNA with the wheel. You're going to get the wrong answers that way. So make sure you're using mRNA. Once that mRNA squirts out the nuclear pore, then the small and large ribosomal subunits find it, clamp down on it like a hamburger, exposing 6 and only 6 bases for tRNAs to come in and match with, right? 2 codons worth of bases for 2 anticodons worth of tRNA. And which letters are on the tRNA anticodon determines which amino acid it picks up. And so that's how DNA, at the beginning, codes for protein, because DNA codes for mRNA, which codes for tRNA, which brings specific amino acids, right? So that's this long interlocking series of matching via shape that ensures that the right amino acids go in the right spot. So the thionine's usually first, it comes in. Then we got our next one, which in this case is going to be leucine. Then you got to form a bond between the 2. That's called a peptide bond. It's always the old thing getting stuck onto the new amino acid. So in this case, it's just 1. In the next case, after the tRNA falls off and the thing slides down 3 bases, the old chain, this is going to keep happening, the old chain gets stuck onto the new amino acid, and boom. That all goes on until we hit one of those 3 stop codons, in which case everything kind of gets snipped apart. The amino acid chain folds up into a protein that can do a job because of its shape, and everything else gets recycled. Questions? That is the central part of this chapter. This stuff is so important. I mean, I told you I don't want to oversell it, but it's like the secret of life. Biologists literally call it, do you know what dogma means? Dogma means like the central ideas of any kind of way of thinking, like often it's used with religion or philosophy. The dogma is like the central way of thinking about something. This is called the central dogma of biology. Like this is kind of the core of everything. None of life on Earth works unless the DNA thing is understood. Yeah.
So with the test, I know you had us draw up the things, but do you want us to know the pictures or, well, we should definitely know what's happening, but do you want us to know the pictures?
I'm not going to make you draw the entire process of translation with all the shapes and stuff. No, I might make you explain it though.
Okay.
I might make you tell me about how, okay, the ribosome clamps on, and then the tRNA brings in one amino acid and another amino acid, and peptide bond. I might have to explain that. You also definitely got to do the math, and it's not math, but like it's almost like math where you have to take, TAC and say that AUG would be the mRNA and UAC would be the tRNA, and it would be methionine, that would be the amino acid. In other words, the stuff you did yesterday and the day before. Okay? No, tomorrow's a regular class. Yep. You've got next week's a full week too. Yep. And then the week after that is ACT. Yeah.
Wednesday.
Okay, and just to review mutations, we said that there are substitution, which will change one amino acid, but then the more serious ones are the ones where you add or delete, insertion or deletion mutations, because they shift the reading frame down and they change every amino acid after the mutation. And then any mutation can basically do three things. Most of the time, and this was a weird number I threw at you, that 97% of your DNA doesn't code for protein. We're going to talk about that today. Rarely it can cause a helpful effect, but of course that's kind of what evolution by natural selection runs on, is those helpful mutations. And then, of course, there can be bad things. So let's talk about that 97%. Now, the first 50% shouldn't surprise you, because remember that there's a coding strand and a non-coding strand in gene expression. Then when transcription happens, the RNA polymerase only runs along one of the two strands. So that basically eliminates half your DNA. A mutation on the non-coding strand wouldn't do anything. But 97 is a lot. And what it means is there's a lot of unused DNA in between genes, separating them. And we used to call that junk DNA. We used to just consider, hey, this is how evolution works. Genes change and parts of genes become useless, and all that DNA you have that you don't use is DNA that maybe your distant ancestors used, but humans don't use it now. And that may partly be true, but it is not true what we used to think, that was all junk. In fact, it's turning out to be way more important than we thought. And we've actually given all that non-coding DNA a name. We call it your epigenome, which your genome is all your DNA, right? Well, your epigenome, that means your DNA on top of your DNA. We don't know that much about what it does yet. But we do know that it breaks a lot of the rules that we thought were true about evolution. We used to always say, there was this famous wrong biologist. How people can be famous for being wrong. A guy named Lamarck is famous for being wrong. He always said, in his evolutionary ideas, he said that, well, the way evolution works is if you're trying for something really hard during your life, then that gets passed on to your children. Like a giraffe is a giraffe because a horse just kept trying to eat the tall leaves, and his neck stretched while he was alive, and he passed that stretching on to his offspring. Now, it doesn't work like that, right? Stretching to eat leaves doesn't change your DNA. And only things that change your DNA can get passed on to your offspring. So Lamarckism, it's called that, and it's a sort of a famous wrong idea about evolution. Except we're starting to find out he was wrong in the way that he said it, but the kind of rule we made because of Lamarck turns out to not be completely true. It turns out that epigenome can get changed by things that happen to you during your life. And as a result of that, the way your genes work can also change. In other words, it is in a limited way, it turns out to, it seems like, be possible to pass things on to your kids because of things that happen to you during your life. Again, we used to think that you couldn't acquire genetic characteristics like that. We are only just now starting to understand that in a limited way, it is possible. The original studies that got us to kind of understand this, I think they're almost exclusively Scandinavian, like in the Northern European countries, because they tend to take very good records in genetics. One of them has to do with a whole town that experienced starvation during one of the wars. And things happened to them because of that starvation that actually showed up in their grandchildren. Like that period of starvation actually caused increased disease risk 2 generations forward, which was unheard of. And so that's what got people kind of looking at the epigenome and looking at, well, what's happening to their DNA when they undergo these stresses to do that. So I know this is a lot, but it's because it's hard to explain. Epigenetics is basically telling us that some changes can get passed on. Turns out that, and it has to do with that 97% of non-coding DNA. Things happen to that spacer DNA that'll turn off or turn on the DNA that is functional. Previous question. Brady.
So if a heavy smoker like smoke like 2 packs a day,
Yeah.
and then would his like grandkid have like major problems?
So no, it's not a direct effect like that. It's like effects that we are hard to even guess at until we see them. But it is things like chemical use, like starvation, like overeating, that we've seen mess with that in between DNA. It's called methylation. It has to do with sticking carbons onto the DNA, and it can either cause genes to be more likely or less likely to be used. And so, if I had time, I'd throw the actual study at you from the starvation thing, because I forget what it caused in the grandchildren, but it was like an increased risk of, I think maybe either heart disease or diabetes or something like that. But no, it's not a direct, like, you know, you smoke all your life and your kid's more likely to be a smoker. You smoke all your life and your kid has lung cancer. No, it's not like that. It's like other stuff. Which is kind of a bummer for folks who kind of think like, well, it doesn't matter what I do to myself. At least I won't pass it on to my kid. Ooh, I don't know. We don't know that for sure right now.
I was gonna say, so like my mom said that alcoholism runs in person. Is that true?
It is absolutely true that there are genetic factors that contribute to alcoholism. Yes.
Addiction. That's
Well, but it's also genetic, right? Like there are genes that affect your brain. It's definitely been shown that high tolerance to alcohol, which is genetic, right? If you're one of those people who can drink a whole bunch and not really feel it, that actually contributes to alcoholism, because of course, if you're socially drinking, you feel like you can have more and you'll habitually have more. But then there's also the actual addiction parts that can be partly genetic as well. Yeah, good question. Other questions? Because I think we're going to wrap it up there. Yeah, right.
mean, when I was younger, a lot of my friends' parents were like veterinarians and stuff like that. So I
A lot of them were veterinarians?
a lot,
That's interesting.
it's a never was years ago. But what I was always taught was like the giraffe. I got this long neck is because all of the other animals were eating the bottom leaves and
Yep.
they just
And so, but it's not, so the difference is,
over
it's, it's not even over that giraffe's lifetime. And we're getting to evolution is next for us, which is great. But the idea is it's not that the giraffe stretched. It's that the animals that had naturally long necks did better because they could eat higher leaves, right? And so they had more babies and passed that gene for a long neck onto their babies, and the long necks became more
It's also like
common.
Yeah, Brady and Eileen was talking. What did she say? Yeah.
You missed it.
Exactly.
Okay. I have two weird things for you to do that I made up completely.
I'm keeping my clothes
Comforting.
on.
Now, two angles on understanding gene expression better. One of them I made up completely. This is the story of the Audi family. Do you remember our analogy? Yes, like cars. You remember our analogy with a cell? What did we use? A city or a house? Yeah, this is kind of like that, but way more complicated. I made up a story. I made up a story about a family that runs a car company, and you have to figure out which bolded parts of the story. Can you do that when I'm not? Oh, you have to figure out which bolded parts of the story correspond to which things that have to do with gene expression. It's going to be kind of a pain in the butt. But if you do it right, it'll mean that you actually understand how this works, right? To do an analogy, you really have to understand how the thing works. That's thing one. Thing two is an idea I stole from my favorite nerd webcomic guy. What he did is he actually made a whole book. I might even have it here. Did I bring the book? I will show you the book when I find it. But basically, it's the idea that if you can explain something in the most commonly used words, like if you can explain something in really simple words, then you really understand it well. And so he made a text editor that only lets you use the 1,000 most common. Yes, but this is a different one. Did I already explain this?
yeah, but it's
We did it with the cell. Yeah, so this is with gene expression. And that was his drawing, the comic guy, that drawing. This is just me trying to explain it in those, limiting myself to those words. And again, you got to figure out what the bolded things are from the same list from the front. You have the same words to choose from. Make sense? Okay. That's what I'd like you to work on for the remainder of the time. Toward the end of the period, I will also give you sample essays, just for those of you who like to get started early. Those are going to be due on, for now, I'm going to say Monday, because the test is Tuesday, and I'd like for this one for you to have those done for Monday so we can look at it. Okay?
When's this due?
That one's due also Monday. Also Monday? Also Monday. Yeah.